Good afternoon, and welcome to the 7th annual Braden Diabetes Fall Fest and Meet the Professor session. I am Linda Gaudiani, the Medical Director and co-founder of the Braden Diabetes Center and Diabetes Care Center at Marin Health. And I am so happy to have you here for what I think is going to be quite a treat this afternoon, a bit before Halloween. We're glad that you're in for this exciting program. A bit of history first. The Braden Diabetes Center was founded and opened its door for the first time in 2014, after about 2 decades of effort. Since then, the center has had some 10,000 or more patient visits and is the major diabetes teaching and management center in the North Bay and now in Northern California. We celebrated our first anniversary in the fall of 2015 with a wonderful outdoor picnic, patient fair, and our first Meet the Professor session. The idea was to make this a unique event which brought together both physicians and patients for a top-level educational talk, and also to get together and share a culture of awareness of diabetes with one another. Our speakers were from the first and continue to be world-class experts, beginning with our program mentor and our godfather, Dr. Earl Hirsch, who has really enabled us in so many ways to pattern our program after his, and that's been most successful. We had other stars, including Dr. Matthias Hebruck, Dr. Salia D., Dr. Robert Rushikoff, Dr. Stephen Kahn, and Dr. Bill Polanski, all of whom we learned from, and we remain grateful to each of them. Since COVID, we have had to change our venue and transition from those in-person times to virtual format, but in some way, this has continued and extended our audience. This year is really very unique for us because we have all worldwide been celebrating the 100th year since the discovery of insulin. And that story of the discovery of insulin is one of scientific brilliance, of daring, of persistence, of vision, and ultimately of gratitude. The first patent for insulin was sold for $1. Bruce and I are incredibly fortunate, therefore, to present as our speaker this afternoon a world-renowned leader in the field of diabetes clinical research, who embodies each of these same qualities. He has been a thought leader in diabetes and has pioneered numerous landmark trials and steered many investigational and academic associations. Dr. Jay Skyler is currently Professor of Medicine, Pediatrics, and Psychology in the Division of Endocrinology, Diabetes, and Metabolism at the University of Miami. He is also the Deputy Director for Clinical Research and Academic Programs at the Diabetes Research at the University of Miami. For 22 years, Dr. Skyler was the chairman for the National Institute of Health sponsored diabetes prevention trial for type 1 diabetes and its successor trial, the TrialNet clinical trials. These were clinical trials aimed at preventing type 1 diabetes or interfering with the type 1 diabetes process. He has had continuous grant support from the NIH for decades. Dr. Skyler is also closely involved with many scientific societies. He served as the president of the American Diabetes Association and the International Diabetes Immunotherapy Group and others. He was the founding editor-in-chief of Diabetes Care, one of the most important journals of the American Diabetes Association. He was also founding editor of the International Diabetes Monitor and is currently senior editor of Diabetes Technology and Therapeutics. You can see why it's humbling to present him today. Dr. Skyler is also the recipient of the coveted 1992 Banting Medal for Service from the American Diabetes Association and the 2014 Distinction in Endocrinology Award from the American College of Endocrinology, as well as the 2015 Distinguished Faculty Scholar Award from the University of Miami. I could go on, but in the interests of time, I want to present and turn the program over to Dr. Skyler. I want to say that when I first, with much trepidation, asked Dr. Skyler to be our speaker, and he accepted, I told him about how we were trying to celebrate the 100th year of insulin this year, and I thought that he might present somewhat of a historical view of what happened around the time that insulin was first discovered. And he thought about that for a minute, and then he said, what I really want to talk about is the future of diabetes. And I realized he was so right, that we want to hear from him about how he sees the future with recognition of the past and the work of the past. What, Dr. Skyler, are we looking at for our future and that of our children? I give you Dr. Skyler. Thank you so much, Jay.
Thank you, Linda. And let me see if I can bring my screen up so that I can show you what my thoughts are on the future management of diabetes. And I'm getting a message that my internet is unstable, so hopefully you'll be able to follow along everything and it won't conk out. But scientifically, we're supposed to list potential dualities of interest disclosures, and I have a bunch of them here, and they've all been turned into the appropriate authorities. I'm going to start by talking about the future of type 2 diabetes. My own personal interests are principally in type 1 diabetes, but I think there may be significant numbers of people in the audience who care a lot about type 2 diabetes as well, and there are really some exciting things happening there too. One is that once weekly insulin once a week instead of once or twice a day. And this is the structure of insulin icodec. This is the insulin molecule before any modifications are made. And what happens is there's some amino acid substitutions that are made to improve molecular stability and reduce the degradation of insulin and reduce the clearance of insulin through its receptor. And then to make it long-acting, the terminal amino acid on the B chain of insulin, B30, is removed. And on B29, now the terminal amino acid, is added, first a spacer and then a icosane fatty diacid. What that does is it has strong but reversible binding to albumin in the circulation and reduced receptor-mediated clearance, so it stays in the circulation longer. And the half-life is about 1 week, and that's achieved by introducing these modifications to human insulin. Another way to do it is weekly basal insulin FC, called BIF for basal insulin FC. This is a fusion protein that combines a novel single-chain variant of insulin with a human IgG FC domain. And that's been used to prolong the action of other drugs as well. And therefore, it's designed for once weekly injection. And it is a selective insulin receptor activator and has a profile consistent with once weekly injection, and can be compatible with either single use or multiple use devices, and can be co-formulated with once a week GLP-1s or incretins. And so it's another way of doing this. And what you see here is, if we compare it to glargine U100, the conventional insulin that we use for basal insulin today, the green up and down every day, you can see it in here, is what insulin glargine does. It goes from a low level to a high level and then back to a low level and goes up and down each day. On the other hand, here you can see insulin icodec, and it goes up and doesn't come down for a week, and then goes up again and comes down during the week. And insulin BIF, you can see in here, remains fairly constant. Once it reaches a steady state out here, it stays fairly constant all the time. So these insulins are suitable for once weekly injection. They're both in phase 3 clinical trials at the moment. And I think they're going to end up getting approved for 2022 or 2023. What's the next thing? The use of incretins for obesity. You've probably seen all the TV commercials that talk about Ozempic and Trulicity. They're incretins. And what you can do is you can raise the dose of semaglutide higher than what it is in Ozempic. And when you do that, this is what you get in terms of weight loss. There's 2 doses shown on here, but you can see over a period of 68 weeks, here is compared to placebo, you might lose 2 kilograms, that's 4.4 pounds. But with the 2 higher doses of semaglutide, you can get down here to where you've lost 10 kilograms or 22 pounds over that period of time. And you can see here that the A1C drops rather dramatically and stays down in the 6s for the group as a whole. Really rather dramatic. Now, the other thing is that this works in addition to intensive behavioral therapy and a low-calorie diet in people who are just overweight, who don't necessarily have diabetes. And you can see again, here the body weight change is a 16% loss over time, really rather dramatic. And the other thing you see is that if you start out on semaglutide, and after 20 weeks, you switch half the people to placebo, their weight goes back up, whereas those who continued on semaglutide, their weight goes down. So we really have some very exciting potential for this. And it turns out that the higher doses were approved with a new name, not Ozempic. They approved with the name Wegovy in August and are now available on the market. Another thing that's in development and proceeding rather rapidly is what we call dual agonist incretins. GLP-1 is one incretin, and the drugs that we just talked about, semaglutide, dulaglutide, that's Ozempic, Trulicity, and others, activate that GLP-1 receptor. But now you can make 2 drugs in one on the same molecule. You can activate not only GLP-1, but another incretin receptor called GIP. And the drug that does that activates both GIP and GLP-1 is called Terzepatide. And it's a multifunctional peptide, which has both the GIP and GLP receptor activation and has a half-life of 5 days, so it can be taken once a week. And in the early studies, it demonstrated significant lowering of A1C and body weight. And here, what one can see is A1C. This is placebo, staying pretty much the same at about 8. And the 3 doses of Terzepatide that were used dropped the A1C over a period of 40 weeks to 5.88, 6.06, or 6.08, really rather dramatic dropping. And if you looked at that, the proportion of people who were achieving an A1C drop of to less than 7 is really quite high, and it shows you the total drop. This is another study. No, same study. It shows you the body weight change with drug. With placebo, their weight stayed the same. Their weights are all in kilograms. And here you can see with the 3 doses, you can get a huge drop in body weight that accompanies this. And here you can see that the average weight loss, depending on the dose, was between 7 and 9.5 kilograms. 9.5 kilograms is what we saw, about the same as the 10 kilogram we saw in the previous study with semaglutide. And so it's really about 21 or 22 pounds, really rather nice. Okay, that's all I'm going to say about type 2, because my love is type 1, and I'm going to use the rest of the time to talk about type 1 diabetes. And I'm going to point out that the ideal therapeutic goals in type 1 diabetes, I show them in these 4 bubbles. They're automated insulin delivery, that's here now. And then in development is prevention of immune destruction, the destruction of the beta cells that make insulin. And so if you prevent immune destruction, you'll preserve beta cell mass, or at least beta cell function. And then the other thing we can do is replace or regenerate beta cells. And so I'm going to start with automated insulin delivery. And here you can see we have on the market already the Tandem Control IQ, which is an automated insulin delivery system, a pump connected with a glucose sensor. Also on the market is the Medtronic 780G pump with a glucose sensor. And pending approval at the FDA, but not quite there yet, is the Omnipod 5-. Here's the pump that sits on the body and the glucose sensor, and you control it with your phone. And I'm going to show you the kinds of results you can achieve. Now, we look at results a little bit differently these days than we used to. We used to just pay attention to A1C. But now, because we're using continuous glucose monitoring, and in this case, in this example, for the 13 days prior, the patient was using their CGM 99.9% of the time. And we have targets here for glucose ranges. And the target range that most people recommend is 70 to 180. And in this example, 49% of the time, the patient was in the target range. 4% was low, 8% was very low, 20% was high, and 19% was very high. And you also get a glucose management indicator. That's equivalent to an A1C, but based on the patient's own blood sugars that are collected. And here it's 7.3%. And the average glucose was 165. And you might say 7.3% sounds pretty good. But if you look over here, that's because you had 8% very low, less than 54, and you still had 19% very high, above 250. I would not say. Using automated insulin delivery with the Tandem Control IQ. And this is 32 days of readings. They use the CGM data 100% of the time. And she was able to achieve a glucose management indicator of 6.4% with an average glucose of 129 and minimal risk of hypoglycemia, only 1% low and 0% of the urgent very low, and 73% in range. And the target is to be 70%. But here's the kicker. Instead of 70 to 180, she set her range as 70 to 150 and got into that 73% of the time. It turns out that it was 90% in the 70 to 180, but that's not the range she was shooting for. So you really can achieve remarkable glucose control with these automated insulin delivery systems. And here's one of the studies that brought them to fruition in a phase 3 study. This is the percent of time in range of 70 to 180. When you're using the closed loop or automated insulin delivery, you can see it goes up above 70 throughout the 26 weeks, whereas there's really no change in the control group, which was still using an insulin pump and a glucose sensor that you could be read, but not with the automated insulin. Proportion of over 80% overnight in range. So you don't wake up low overnight. That's crucial. You're getting to be really well done. And even during the rest of the day, you do superbly well. Here's the A1C during the study. It didn't change with the control group, but it dropped nicely with the automated insulin delivery. And here you can see the percentage of time below 70 really drops very, very low, practically nothing below 154. But you can see it's only about 1% or a little more throughout the study, less than 70. And so remarkably good results are achieved with these automated insulin delivery systems. Okay, that's automated insulin delivery. What I've spent most of my life doing, at least the last 35 years of it or so, is trying to do immune intervention to preserve beta cell mass or function. And the reason we do that is this is the natural history of type 1 diabetes in a cartoon originally developed by the late George Eisenbarth, which says you're born with your beta cell function intact and potentially a genetic predisposition to type 1 diabetes. Actually, more accurately, I should say, where the genetic predisposition outweighs genetic protection. And then we think an environmental trigger, we don't know if that's a virus or something else, starts the immune response, and you get a cellular immune response that attacks the beta cells, causes beta cell injury, and what we call insulitis, an inflammation around the insulin secreting cells that damages them. And as they're damaged, they expose part of their cells to the immune system, and the immune system reacts to that because they're not supposed to be exposed by forming antibodies. And these autoantibodies, ICA, which we find antibodies to GAD, to IA2, and the zinc transporter, they tell us that the immune process has begun and that we really ought to follow these people carefully. And you can see there's a decline in beta cell function during that time. And eventually you get to a phase where there's dysglycemia, an abnormal glucose level, or the loss of first phase insulin response if you give an intravenous glucose challenge. We used to call that prediabetes. And then when you had the clinical onset of type 1 diabetes, that was full-blown type 1 diabetes. And you still had a decline over time past that, where eventually you may run out of any of your beta cell function. Well, what's been recognized over the last few years is that may be a slightly off-base way of classifying the disease. And so we've changed the classification in 2015, and we now say when you have 2 or more of the autoantibodies that I mentioned, we call that stage 1 of type 1 diabetes. And when you get to dysglycemia, we call that stage 2. And when you get to symptoms and what we used to call the onset of diabetes, we call that stage 3. We also have pre-stage 1. Those are people who have a genetic predisposition. And what we try to do with interventions, we can theoretically intervene back here in those who are only genetically at risk, and that we would call primary prevention. We prevent them from even going on to stage 1. Or you can intervene during stage 1 or stage 2, and we call that secondary prevention. They've already started the disease process, but we're going to try to stop it from going further. Or we can intervene in stage 3, that would be tertiary prevention, or intervention, some people call it, and that's to try to preserve the remaining beta cell function so it doesn't keep going down. Now, Linda was kind enough to mention that I spent 22 years chairing NIH clinical trials networks, the diabetes prevention trial, what we call DPT1, followed by TrialNet, which began here. And these are the studies we conducted during the time that I was the chairman. We tried to intervene to prevent the disease back here and continued to do some prevention trials in here. As well as studies in new onset disease. We did some mechanistic studies to try to understand things better. And we also are doing some long-term follow-up. And we screen people to get into any of these in what we call pathway to prevention. And I'll talk more about that in a couple minutes. We could intervene with a number of these interventions. And what we would find is it looked like they worked for a while, and then there was a decline in parallel to the comparison or placebo group. Even here, where there was a bump up, there was then a decline. And here, although they stayed different from the placebo group, they were declining all along the way and not really preserving things well. They did better than the placebo, but not the way we'd like them to do. So let me show you the most promising recent studies in this regard. This one is one that uses low-dose thymoglobulin, or ATG. ATG is the abbreviation for thymoglobulin. And that's seen here in the blue line at the top compared to the red line placebo in the bottom. And what patients got in this trial was 2 days of treatment at the time they were started, and then no more. That's all they had of the ATG. And you'll notice that a year later, they're still preserving beta cell function. It practically didn't decline at all. Here's the placebo declining. We tried also to combine it with GCSF because there were earlier studies that suggested that might enhance the benefit. And you can see here, it did not enhance it. In fact, it was not as good as the ATG alone. And if we continued out to 2 years, the trends continued. And although now there was some decline in the ATG group, it was still highly different than the placebo, which declined even further. And, so it's a remarkable way of preserving the residual C-peptide, the way we measure, which is how we measure beta cell function. Now, here's another study. This is the TIGER study reported last year. It was an industry study, not a TrialNet study. It used the anti-TNF golimumab. You've probably heard of Humira. That's also an anti-TNF. And you can see here, it looks like it really preserved beta cell function really quite well during the first year. And then what did they do? They stopped the drug. And when they stopped the drug, you can see it declined parallel to the placebo group. And I'm not sure why they stopped. It's an interesting question. The regulatory agencies sometimes make people stop because they want to see if there's a continuing effect. But I think that's probably the wrong approach. That, anti-TNFs have been used for many, many years, a decade or more, in people with rheumatoid arthritis. Why stop them if they're working in type 1 diabetes? So I think that, there's lessons to be still learned here. Here's another study. This used a drug, anti-IL-21, which was to try to stop the immune response, as well as liraglutide. Liraglutide is one of those incretins that I mentioned before that improves beta cell function. I mentioned them in terms of type 2, but here it's being looked at type 1 together with a drug to stop the immune response. Cell function lower than the starting point, but not much, and certainly different than placebo or different from either of the two drugs alone. But again, because of discussions with the regulators, they stopped the drug. And you look what happened. Goes straight down, and ends up matching the placebo at the end of the next half year. Again, I don't think it should have been stopped, particularly not the liraglutide, because that's improving beta cell function. You take away something that's improving beta cell function, it's not surprising that it declines. Here's another drug, another study. This one was led by Steve Gittleman from UCSF. He used imatinib. Imatinib is a tyrosine kinase drug that was given, in this case, for 6 months. It's a great drug for leukemia, and there was suggestions that it might work for diabetes. And you can see that at the end of the 6 months, it was different than the placebo, but then they stopped it and it continued to decline. Not as fast as placebo. It got its kick here to move it up higher and then persisted a bit, but still, had they continued it longer, and you can do that in leukemia, success story that we've had. This was a study we started in TrialNet while I was the chairman. It uses teplizumab, an anti-CD3 monoclonal antibody. Patients who were enrolled in this had stage 2 diabetes. They hadn't developed full-blown diabetes yet. And the treatment was 14 days of treatment at the time of enrollment, and then no more. And what you can see here is if you follow them out and you compare the placebo group to the teplizumab group, at the time when 50% of each group ended up with diabetes, there's a 32.5 months delay. You prevent the evolution to type 2 diabetes by nearly 3 years. And so you end up with 50% and 50, because that's what we're looking at here when we stopped it, 50-50 of stopping it with teplizumab, and only 22% stopped, were free of the disease with placebo, and the other 78% had gone on to develop the disease. And so that's a remarkable finding, and it led to a submission of teplizumab to the FDA. And at the FDA Advisory Committee in May, Kevin Harold, who led the study, presented the data and said that, teplizumab, as given as a brief treatment, 14 days, was safe and had long-term potential benefits. And at 5 years, 18% of the teplizumab patients did not have clinical diabetes, as opposed to 6% in the placebo group. So it's the first drug to demonstrate preservation of beta cell function, leading to delay in onset and potentially, in some patients, the prevention of clinical type 1 diabetes. So more studies going on, and hopefully the FDA will ultimately approve this. Right now, they're trying to get some additional data on manufacturing issues. But you can participate in these trials, either new onset or prevention trials, by going to the website, which is down here, https://www.trialnet.org/participate. And people who are eligible to participate are first-degree family members between the ages of 2.5 and 45, second and third degree family members up to age 20. And it involves a small blood sample where we test for those autoantibodies and then do the other things, if necessary, to follow you up if you have positive antibodies, to see where you stand. And in new onset diabetes, you can just directly enroll. There's no fee by this. If you go to the website, because it's funded by the National Institutes of Health. And those of you who are here in the California area, the nearest clinical center for TrialNet is at UCSF, and karen.co@ucsf.edu is a way to get a hold of her if you have more questions about this. The website gives lots of good answers too. Okay, that's immune intervention to try to preserve beta cell mass or function. The next thing I want to take up is replacement of beta cells. And beta cell replacement has actually been around for a while. Pancreas transplants have been around since the late 1970s, early 1980s, usually accompanying a kidney transplant. If you're going to put somebody on anti-rejection medicines to prevent a kidney rejection, and if you can get the pancreas from the same donor, why not give both organs? Was the argument, and it often worked quite well. It's big surgery in terms of the recipient. The donor is deceased, so the donor doesn't care. But it's big surgery, but it does work. But since it is big surgery, the question is, could you get away with less surgery by doing islet cell transplants? The islets that include the beta cells that make insulin, the alpha cells that make glucagon, and you can isolate and purify them. And that's now usually used for people who have recurrent severe hypoglycemia, resulting in seizure, coma, or admission to the emergency room because of severe hypoglycememia. And if you've had that a couple of times, hopefully you've learned to try to prevent it, and maybe doing that with automated insulin delivery. But if you have that, those are who we generally use islet cell transplantation for. Now, the problem with both of these cellular replacement approaches, either the pancreas or the islets come from organ donors. Those are often motorcycle accident victims. That's the largest single source, particularly ones who drive without a helmet. But there's only 1,200 pancreases which become available from organ donors in the entire United States each year. And you probably have more than 1,200 people with type 1 diabetes who just come to the Braden Diabetes Center. So, it's not going to go a long way. So what we need is a better availability and source of cells so we can have a more unlimited supply. We also want to try to deal with the problem of rejection. so that they don't get rejected. And I'll talk about ways to do that in a minute. But the other thing that's come up, particularly that we've been able to observe in people who had pancreases and kidneys, is that you can get recurrence of the initial autoimmune response in the pancreas. And we can tell it's not rejection because we can biopsy both of the organs. There's no signs of rejection in either one. Kidney function is working fine, but pancreas function is decreasing and diabetes is recurring. That's not what we want either. And so we got to figure out a way to overcome these problems. Well, what's the way to overcome the source of cells? There's two possible strategies. One is xenotransplantation using pig islets. And there are now colonies of pigs that are raised to be free of pig viruses so that the organs can be suitable for human transplantation. Xeno is means a cross species. Or we can use stem cells. And there's several different stem cell strategies. One strategy is a patient-specific strategy. You would take a liver biopsy from a patient, take it into the laboratory, and take those liver cells and de-differentiate them so that they are potent to go in any direction. And then you reprogram them, or if you will, you trans-differentiate them into insulin-secreting cells and into islets. Problem with that is that you need to have a laboratory capable of doing that in a reproducible way for that patient and the patient has to go to that particular site to have that done. And that is not necessarily easy to set up or convenient for patients. The other thing we can use is allogeneic cells. Allogeneic means that they differ the same way you would get from a donor of a pancreas or kidneys. And you can do that with a bank of embryonic stem cells. Human embryonic stem cells is the abbreviation, or with induced pluripotent stem cells. And here's the abbreviation. And those you get by taking tissue from a person and converting centrally for companies to get into the game of trying to use these kind of cells as a source for correcting type 1 diabetes. So how do we deal with the immune protection? Well, you can either give immunosuppressive or immunomodulatory drugs. That's what we do now with pancreas and islet transplantation. Or you could create a physical barrier. If you will, you encapsulate the cells to keep the immune cells that are targeting them from attacking them because they can't get into the capsule. Or you can do gene editing for immune evasion or immune protection. You in the Bay Area are familiar with Jennifer Dauda from UC Berkeley, who won the Nobel Prize for introducing the CRISPR technique of gene editing. We can think about doing that here. I'm going to show you two strategies. This one is by a company called Therapeutic, started from Harvard Laboratories, and then was bought by Vertex, a bigger company. And they have a clinical trial using induced pluripotent stem cells. And the key inclusion criteria for their trial is a clinical history of type 1 diabetes of at least 5 years duration. In the last 12 months, 2 episodes of documented severe hypoglycemia. Remember, that's seizure, coma, loss of consciousness, admission to an emergency room. You have to be on stable treatment and continuously using a CGM, continuous glucose monitor, for at least 3 months and throughout the duration of the study. And if you've had a prior transplant of any kind or cell therapy, you cannot participate. And there's other exclusion criteria as well. This study has been underway since April. They're having trouble finding patients to enroll because there's many less people now having recurrent severe hypoglycemia because we use automated insulin delivery a lot. Now then, ViaCyte is a company in San Diego, and they have a different set of product families that they used. They started out using what they call PEC and CAP. And the cells they use in all of these are PEC-01 cells. And they went into clinical trials with this, with a device called an Encaptra device. That was how they encapsulated it. And that would mean that the blood vessels only get to the surface and have to diffuse in and get oxygen in and nutrients in and then insulin out. And because they're encapsulated, they don't need long-term immunosuppression. But what they found was they were getting a foreign body reaction that was making these cells, these devices not work. So then the next study they did, they went to what they call PEC Direct. They put holes in the capsule. So it's an open device. And now they could have direct vascularization. They didn't get the foreign body response, but they need long-term immunosuppression. And so they've been using that in high-risk type 1 diabetes, and they've reported some promising results. They're now working with the Gore company to get a newer device, which hopefully will not have the foreign body response, so they could go back to the encapsulated approach. And that would be potentially useful for all type 1s and insulin type 2s. And in the long run, they're working on, and still in the discovery phase, what they call PEC-QT, where they're using the immunoevasive techniques, CRISPR, so they can use an open device, direct vascularization, and they don't expect they'll need long-term immunosuppression because they've already created immune evasion and immune protection. So those are how they're proceeding. Let me tell you about this immune evasion. To overcome the molecules on the surface that are of cells that are responsible for the rejection, we call HLA molecules, class 1 and class 2. And what you can do with CRISPR is knock them out, so that then you have the cell evades the immune system because it doesn't have these markers on it. Alternatively, or in addition, you can put in things in the cell which will create immune protection, force away the other molecules. This symbol means turning them off, that T cells, natural killer cells, and macrophages that might cause that. Which will work, and there are challenges. Challenges for any of these are to be able to scale up and reproducibly manufacture product. That is no easy task. And that in that manufacturing process, you need to have good cell cultures and preparations to make enough of the cells to be able to be used, and then you've got to be able to deliver them. That has not been easy and has kept progress being much slower than we'd like to see. So I'll remind you of my ideal therapeutic goals. Currently, automated insulin delivery. Prevention of immune destruction to preserve beta cell mass or function is in development. And hopefully we'll have teplizumab approved for slowing development of the disease sometime in the next year. Replacement of beta cells. I didn't really talk about regeneration because it's even earlier in research process. So what it takes to make everything work is a breakthrough and innovation, how you put all these things together. And if you have the corner of breakthrough and innovation, Breakthrough Boulevard and Innovation Avenue, that's been labeled Hope Junction because it gives us hope that we'll be able to tackle diabetes. This is a Boehringer Ingelheim of the US, but when I saw it, I had to have a slide because it really does express what we're trying to do. And so I look forward, I'm an art collector, and I look forward to the future through the eyes of one of my favorite artists, the Belgian surrealist René Magritte. And as I look into the future, I can see where we will get to a world without type 1 diabetes. Don't ask me how long it'll take, because every time you turn the corner, you discover new problems and new things that you need to deal with. But hopefully we'll get there. Thank you for your attention.
Wow. Thank you so much, Dr. Skyler. That was amazing. It is hopeful. It is also really complicated. But there's been so much progress. And I think that's where today we stand when we think that 100 years ago, type 1 disease, or 110 years ago, was a completely fatal disease to children and young adults. And now those same people lead completely normal, productive, even basically many of them completely normal longevity to their lives. That probably seemed impossible 100 years ago. But thanks to people like you and you, we are at a different crossroads that they perhaps could not have quite imagined. So you've given us a lot to think about, certainly. I want you all to meet the panel that we have brought together today to ask Dr. Skyler some questions. We had already created some questions around this topic, but I would welcome others to either come in with a chat question, or if anyone in our panel has an additional question after listening to Jay's talk, that we should go ahead and open up the floor for those. Let me briefly just introduce, however, our panelists today. I want to start with introducing my dear friend and the co-founder of the Braden Diabetes Center, named for Bruce Braden, who was so instrumental in supporting my early efforts to bring a diabetes center to our health system and to Northern California outside of the university settings that we also depend on. Bruce has been a very successful person, a very successful consultant to the Braden Center that's just continued to grow. And thank you, Bruce, for being with us this afternoon. I want to then introduce Jessica Oliveira. Jessica is our inpatient nurse specialist at Marin Health Center. And the fun story about Jessica is, although she was an experienced nurse already in another field, she became interested in becoming a nurse specialist in diabetes, and she entered into our sort of unique Braden Diabetes student internship program in 2019 and was our intern for that year, and did some amazing work with the other nurses. And thank you, Jessica, for bringing that professional experience, as well as your personal experience. As you have told me, it's okay if I share this history that you have been living with type 1 diabetes for 27 years and are quite passionate about advancing diabetes technologies and treatments and education. So you'll have some questions for Dr. Skyler. And last but not least, my sidekick in most crime, Kelly Stroop, who is our clinical nurse manager at the Diabetes Care Program and at Marin Health Center. And it was a lucky day when Kelly joined our health system more than 4 years ago. She has had personal and professional experience in diabetes over decades, and she is now our department manager, as well as serving on our inpatient glucose management team. She was diagnosed with type 1 diabetes also in 1985, and that has not stopped her from a stellar professional and personal and family career. And I'm happy to say that she received the first ever Braden Diabetes Outstanding Achievement Award this year for her critical role in initiating our first virtual BDC Fall Fest last year, and also for all her help as we went for Joint Commission accreditation and received that accreditation this March as a hospital for advanced diabetes management with our inpatient service. So that's our panel. We're going to start with questions right away because we don't have too much time. I think, Jessica, you were going to ask Dr. Skyler of the question that you first had.
Yes, I do have a question. So since I've had diabetes, there's been so many amazing advancements in the medications and the technology. It just seems to get better and better. I'm wondering if you see in the future that it'll be as simple as an insulin pill or just a once a day oral tablet that we can take and move on with our lives.
Well, there are some oral insulins in development. I have not been impressed that they're ready for type 1 yet. I think they'll be, if they succeed, they'll first be for type 2. You can't really get the peaks that you need to control meals for short-acting things. And, you know, I think that for long-acting, closer to the market is the weekly insulins that I mentioned for basal insulin, which I think could be useful for type 1 as well as type 2. And both of them are going to be studied in type 1.
Okay, that's important. I think that's something that people don't always understand, why you can't take insulin in an oral form. I want to move on to another question that grows out in part of all the data that I see in the trials that are ongoing, and the fact that we also do clinical research in my practice and have for some 38 years. And the basic question is, how do you think it is possible to present these newer strategies in research stages now? How do you present them to your patients for participation? How do you think we as doctors can balance the benefits as well as the risks for a patient to participate in a clinical trial? You must face that all the time.
Oh, just this morning I was talking to a family, and you know, they wanted to just be sure they got drug and not placebo. And you know, one has to point out that until something has been through a clinical trial and been shown to be beneficial, you may be better off if you're randomized to the placebo group, because the people in the experimental group may have a side effect, or it may not work. And you know, hopefully by participating in the clinical trial and your contact with the clinical center, you know, you really do get your care optimized. And so you'll get ordinary care optimized, and the potential that if the treatment that you're trying to test out works, you may benefit. But you have to balance risks and benefits. I've had many trials where I only had 10% of eligible people who enrolled because they were concerned about the risks or about the burden of going through the procedures that were necessary to participate in the trial.
Right. And here again is where we're at a crossroads when we hear the stories of the first development of insulin. And we know that some of the scientists themselves took crude preparations of insulin early on before they even knew that it could work, and it could have even hurt them, and probably did. And yet this is sort of the push of science, that without this research, we just can't discover the next truths. So you face that, we're facing that too, but I think it's helpful for we as clinicians and for patients to realize that without research, it's difficult to move forward in these complex scenarios. Kelly, I know you also had a particularly a professional and personal question to ask Dr. Skyler.
Yes, definitely, Dr. Skyler. I love your presentation. It's very interesting, and since a patient of type 1 diabetes, passionate about what's happening kind of behind the scenes that we don't see until it actually comes onto the market. But you talked about the actual stages of diabetes, and where are we at with actually the preclinical stages of tracking and prevention of total pancreatic beta cell loss to complete insulin resistance? As far as myself living with type 1, having children, you know, where are we at with diagnosing that? Are we, you know, what are the recommendations? Do we test families for those risk factors? Are we prevention of is kind of where we're headed, I think, with a lot of the research that you're specifically doing for type 1. So looking at that, how close are we in diagnosing preclinical stages?
Well, we can certainly diagnose it now if people have tests. And there has been to be screened. TrialNet has specific criteria.
Can you repeat that? You froze up. Jay, you froze up for a minute, and that was so critical what you were saying.
Okay, I'll go back and say it again.
Thank you.
Yeah, no, I think that anybody who is concerned ought to be screened. And if you have specific criteria, I mentioned the age range and being a relative, you can be screened in TrialNet, but you can be screened through there's a JDRF website that allows you to be screened if you're just anybody who's interested in being screened. Ultimately, I think what we're going to have is something that's being done in many parts of Germany already, also in Colorado, is screening all babies at birth for the genetic risk of type 1 diabetes. And then whether or not you ought to be tested for antibodies and the other things, or there may be some potential clinical trials of people who are just genetically at risk who haven't yet developed stage 1 to see if you can stop that. And so I think that's sort of the next kind of wave of things that we're going to see more of.
I know that Jessica has a question. It sort of straddles type 1 and type 2, but Jessica, maybe we should go to that first, and then I'm going to wind around to another specific type, more of a type 1 question.
So I have, with type 1 diabetes, been off and on following a ketogenic diet, which I know is becoming more and more common for people with diabetes. Also intermittent fasting. I'm wondering how you feel about the safety, long-term risks, benefits, and then specifically safety with a ketogenic diet for someone with type 1 diabetes versus someone with type 2 diabetes. And a lot of our type 2s are taking SGLT2s. I'm wondering how you feel about the safety of a ketogenic diet and SGLT2s as well.
Yeah, these are complicated questions.
Yes.
What was found when SGLTs were tested in type 1 is that they did allow people to decrease their insulin dose and achieve some better targets, but at the risk of DKA. And ketogenic diets in type 1 would also carry a risk of DKA. So one has to be particularly cautious about monitoring ketones extraordinarily carefully in type 1 if you're going to go on a ketogenic diet, and realize that the DKA that you can develop with either SGLTs or with ketogenic diets, the glucose may be normal while the ketones are high and lead you into ketoacidosis. And so I have a great deal of concern about that, and personally have not recommended ketogenic diets or SGLTs in type 1s because there's a lot of work required to keep up with the ketones and keep up with the insulin dose. On the other hand, ketogenic diets in type 2, particularly people who are not on insulin, can be dramatic and can actually lead to profound weight loss and even remission of their diabetes. And so that can be really very, very good. And in prediabetes can lead to prevention of type 2 diabetes by helping people lose weight and get better control of things. So the answer is a little different depending on the circumstances. And patients with type 2 who are on insulin, it really depends on how much insulin they need and whether it's possible to decrease some of that if they're on a ketogenic diet without getting into trouble. But you still have to monitor your ketones carefully. there's not a simple answer for your question.
Thank you.
Dr. Skyler, just as a trailer to that question, it seems like we have sort of pretty fantastic glucose monitoring and pretty klutzy ketone monitoring. And do you see a device coming down the pike where patients would be able to monitor their ketones more easily than by having yet another monitor, which is less accurate perhaps than one would like? Where are we going?
Ketones is still to some degree in the dark ages. And I hope that we're going to get out of the dark ages at some point.
Okay. All right. Just the devices aren't there yet, but they're in development.
There are devices in development, but at the moment, not ready for prime time.
Mm-hmm. Kelly, your two questions kind of got asked at the same time, so I'm going to.
So there's some questions on the chat.
Mm-hmm. Do you want to pull those up, or should I have a couple of others, but if there's something that looks good on the chat, I didn't bring it up. Kelly, do you want to look at them?
Well, the first one, I think, Dr. Skyler, would you mind for our audience, what is an SGLT2?
Sure.
Thanks.
SGLT2 stands for sodium glucose transporter 2. And they're drugs, there's several of them out there: empagliflozin or Jardiance, dapagliflozin for Xiga, and canagliflozin, Invokana, that have been out for a while, and they're used to treat type 2 diabetes. And one of the remarkable things about them is that they've been shown to not only decrease the risk of heart disease in patients with type 2 diabetes, but profoundly decrease heart failure in people with or without diabetes, and they slow the development of kidney disease. So they really are a great group of drugs to use in the properly selected patients with type 2.
Thank you. And I think, Dr. Gaudiani, there is another question in the chat. And I actually, because mine were kind of lumped in together, had a question really specifically for personally, but also for working on the inpatient setting and maybe for some of our providers that are joining us. Once weekly basal insulin that could potentially be used for type 1, what is the risks of titration for both? Maybe their patient's going to be NPO or they have an unexpected emergency, and they have a half-life, an active drug half-life that's going to last quite some time, in the titration of that and the safety of that for patients that are coming into the hospital.
No, that is a concern, because they'll have insulin on board, and if they have to be NPO for a procedure or for something, you really need to be sure they're getting IV glucose, and you're monitoring them extremely carefully, and watching them. There's no simple answer to that.
And I'm sure one answer is going to be another futuristic thing we don't have right now is continuous glucose monitoring in the hospital setting. I mean, we could have it, but wouldn't that make a huge difference?
It's coming. A lot of places used it during COVID, because they couldn't get to the patients regularly to get blood glucoses.
Right.
It's not yet approved for use in the hospital routinely, but most of that is because the FDA requirements are that one needs to demonstrate that there's not any interference with many of the drugs that are used in the hospital. And therefore, you got to go through a whole slew of drugs to demonstrate that they don't cross-react with the glucose monitor and potentially alter the readings. We know that hydroxyurea does, for example, but that's a rarely used drug except in thrombocyte diseases in hematology. So it's not a big deal for most patients, but it can be, and there may be others as well.
Right. I can certainly see a day coming up pretty soon where as patients come in, they won't just get put on a cardiac monitor and an oxygen monitor and a blood pressure monitor. They'll be put on a continuous glucose monitor, maybe even if they don't have diabetes because of stress-induced.
Absolutely.
Yeah.
many people consider glucose levels another vital sign, and therefore you need to know what it is.
Right. It's sort of interesting. One would not have thought about that a decade or two ago. Something that really is plaguing me, and I am disappointed at this. We know that we're experiencing an increase in both type 1 and type 2 diabetes in many, perhaps not all countries worldwide. It seems that we have more answers to the etiology or the causes of why there's more type 2 diabetes, attributing it to obesity, dietary and nutritional changes and deterioration in choice or availability of foods that are healthy, reduced physical activity, and more stress. But what are your thoughts about the causes of the increase in type 1 diabetes? And there, could this be reversed? When we were going to school some years ago, it was thought to have something to do with mothers not breastfeeding. Now we have a generation or two, at least, of women who have breastfed, and yet the incidence of type 1 diabetes, I understand, is going up, at least in our country. And I think it was Dr. Hebrook that told me, but it's really quite rare in Africa, for instance. What is going on with the increase in T1D here?
Yeah, I can't completely answer that. But one hypothesis that's used is what we call the hygiene hypothesis, that when you're exposed to environmental contaminants, pinworm, for example, or various infections, it activates the immune system in such a way that it protects against autoimmune diseases, because you build up a stronger immune system that can turn off the attack. And that some people have said that's why you don't see it in Africa, because they have a much dirtier environment. All the kids end up with worm infections and other things early in life. And in some tribes, they actually feed the mother's feces to the baby, and they have lots of different infections. And that may be bad initially as the infection, but it may protect you against autoimmune diseases such as diabetes and asthma. And so, that's a hypothesis that has been around for a while, championed by a number of people in Europe in particular. And
I think we've got you frozen again. Go ahead. Dr. Skyler, you froze up on us a bit.
And when they got together, the German cleanliness from West Germany spilled over because they became more economically viable. And the frequency of type 1 diabetes in East Germany rapidly caught up with that in West Germany.
Interesting.
And so, the hygiene hypothesis is one theory. There probably are others as well, but I can't give you a firm or good answer to the question.
That's a little bit scary, since we're all living with bottles of antimicrobial soaps next to us all the time since COVID. Right.
We have to protect against COVID. It's a more immediate problem.
Absolutely, of course. Of course, I didn't mean to suggest anything other, but it's the consequences of each onslaught is hard to predict. Kelly, do we have any other questions that came in? Did we cover everything here? Oh, I guess I had one more, Jane. Maybe we have just another minute or two before I wrap things up. And I was going to ask my question in two parts, and the second part is really the one that I'm the most interested in. But looking back over this illustrious work of our forefathers and probably mothers over the last 100 years since insulin was discovered to be a life-saving therapy, what do you think was the single most significant subsequent development in insulin therapy between its discovery and now? And secondly, going forward and looking at—
I missed your first part of the question because the internet froze here.
Oh, I'm sorry. I was going to say, what do you think, since the first development, the discovery of insulin up until now, so many milestones, what do you think was the single most impactful milestone in insulin therapy since then? And going forward, what do you see as the most promising and possible life-changing insulin therapy in our future. Obviously, we have complete eradication of diabetes is our goal, but prior to that.
I think the insulins today are extraordinarily better than, they've been progressively getting better over time. They're pure. We switched from animal-derived insulins, where the FDA insisted that every batch of insulin be tested in rabbits before it could go out. And now we have, automated procedures where we make it by recombinant technology. And so, insulins have gotten a lot better. And we've had designer insulins, so that we have basal insulins that are truly flat and peak insulins, which come on very quickly to control meals. And so insulin therapy has gotten a whole lot better. In terms of insulin therapy, it's been so progressive in so many things, I can't point to a single thing that has done it. But in terms of lives with type 1 diabetes, the other single big thing that's been important is the introduction of continuous glucose monitoring.
Right. It's really, like what most people tell me, it's a game changer.
Yeah, it is the game changer.
It's the game changer.
Along those same lines, Dr. Skyler, there's a question in the chat, both on CGM and the technology of on CGM reading to a Fitbit or another application potentially where the patient doesn't actually have to have their phone on them. And that's not, necessarily reading all the time, where somebody can just be wearing their Apple Watch or their Fitbit. What do we know where the status is on that type of technology?
It'll be available on the Apple Watch next year.
I hope our attendee is jumping for joy on that one. That's pretty quick compared to what they expected, I think. We just have a couple minutes left, Dr. Gaudiani, if you have another question, and then we'll let you do your final remarks and comments. Maybe we should allow Bruce to ask a question. He's been quite silent in the background.
That's what I was thinking. Bruce, how is this structured with your experience?
No, it's, we've been talking with this before, and I've talked with some of my friends. The progress, I had diabetes, started about 36 years ago, back when you tested your blood with what we called the guillotine. And now, I've got a monitor and I can swipe it at any time. That's the biggest difference. The syringes have gotten smaller. I'm still old school, and I still use a syringe and insulin. I keep thinking I'll change that at some point, but to be determined. Anyway, it's the progress in the treatment of this disease, and I've got some friends, and I said, you know what, if you'd better kind of listen up, because the best way to control this disease is through education, and that's what we're getting today.
Well, I think nothing could sum it up better than that, Bruce. Thank you so much. And thank you so much, Dr. Skyler. You've covered so much territory. I am so glad you prompted us to go look at the future, even as we acknowledge the past. And I just can't thank you enough for the time you've spent with us. I know you often speak to much bigger groups, but I think this will have a huge impact on our community. You are one of us now. Thank you so much.
Happy to have been here. Good night.
Good night. And I want to say thanks to some other folks too who made this evening possible. I really want to extend a true thanks to our CEO, Dr. David Klein, who's been with us just about a year now. But since he came on, he has just been so terrifically supportive of the expansion of our diabetes service line, our growth, and our anticipated milestones. So thank you, Dr. Klein. And to the entire staff at the hospital, our colleagues, our nurses, and to our staff at the Braden Diabetes Center and the inpatient for doing such a terrific job and celebrating this year of 100th year of insulin with us. Special shout out to Kelly and to our administrative assistant, Julie Roddy, without whom we could not have thrown this together and assembled it as smoothly as it was. And thank you so much, Kelly and Julie, for your dedication. I want to thank all the work that is done by the Marin Health Foundation, which Bruce is the president of this year, for our hospital development and for our medical center. And to our community diabetes champions who support us financially and in other ways and set the example of how the community can step up and make things better for the place that we're all living and sharing. I want to thank our patients whose courage and cooperation and perseverance is the thing that I think inspires all of us. I want to say that before we decided on the future as our topic, I had spent some time looking over slide sets about the history of videos about the history of insulin's discovery, and it was very inspiring. I've read so much about that. But at the end of the day, I was also saddened that amongst all of those pages, I saw hardly any pictures of women. And I was reminded that for so many decades, and for this past century, there was so much barring women from participating at that professional level. That's changed now. If one goes to the meetings, there is a sea of women, physicians, endocrinologists, certified diabetes educators, nurse practitioners, CDEs, people that are sometimes referred to as mid-level. I think that implies a hierarchy that is totally inappropriate. They are front and center, warp and weave of the treatment and the future care of diabetic patients. And I want to thank them. And I think I want to thank and reach out to a missing group of women before these women who are now in medicine, myself included, and all the others coming aboard. For that generation or two, there were women who had children with diabetes, with very few supporters, with very little in the way of monitoring, who had to stick their kids with huge needles, who had no way to test their blood, had crude testing of urine only. Many of those women, such as the mother of our esteemed colleague, Earl Hirsch, who raised two little boys in the 60s, back to back with diabetes. And they got out and not only took care of their kids, but they made a difference in the development of the American Diabetes Association, the JDRF, and other associations. I know that Gloria Hirsch was instrumental in starting the first ADA marathons and walkathons and summer camps. And both she, as representative of probably hundreds of other mothers and fathers, but especially mothers, I think, who took care of these kids until better things came along, as they did, as you've just heard. So I want to shout out to those mothers. I want to shout out to the mothers and fathers today that are dealing with this and say there really is, as you heard from Dr. Skyler tonight, a huge amount of hope as we move forward. And we just encourage you all to keep at it, keep working with us. We know that there is going to be a very bright future that continues to unfold. I want to thank you for your attention tonight and wish you all the well in health as you go forward this year. Thanks so much. Good night.
Thank you very much, Dr. Gadiani. So a quick reminder before we close for our attendees, if you will be requesting continuing education credits for the event tonight, please email us at diabetes@mymarinhealth.org. And also, if you're interested in partnering with us to support the Braden Diabetes Center, you can contact Noelle Jew at the Marin Health Foundation at 415-925-7002. Also, you can feel free to contact the Brain Diabetes Center at 415-925-7370. Thank you very much.